Good evening, ma'am.
Good evening, sir.
Good evening, ma'am. Good evening, Varsha. Good evening, ma'am.
Sir, you can start now, sir.
Yes, ma'am. Thank you. Good evening, my dear students.
Good evening, sir.
Last class, yeah, Varsha as well as Dhanalakshmi. You are all are warm welcome to the once again the biology classes. Today, what we are going to discuss, about DNA replication. You know very about the DNA of the genetic material, already you know very about. But DNA itself, how it can replicate. As a result, what will be the end product? Any idea? Replication. Dhanalakshmi.
It's a copy of DNA.
Very good. Simple term we may call copy of exact copy of the parental DNA, isn't it? See here. So the DNA replications. The DNA replication is the process by which a double stranded DNA molecule is copied to produce two identical DNA molecules. See here. So this is the DNA molecules, double stranded helical structure, and parental DNA. It produced a exact copy of DNA. How many copies? 2 copies. Okay. 2 identical DNA molecules produced from the replication process. How it will be happen? What are the enzymes involved? What are the basic process is there? That things we are going to discuss one by one very deeply. Okay. See here. What is the main uses of the replication process, my dear? What is the main uses? What is meant by PCR?
The polymerase chain reaction.
What is the use of that one? Where it occurs? Where it occurs, my dear? What about Varsha?
So it is used in DNA fingerprinting.
But replication is used in DNA fingerprinting, or is it? Is it correct?
Yes, sir.
Okay, anyhow, fingerprinting the DNA replication used on. Okay, anyhow, the DNA replications mostly used for polymerase chain reaction, simply called as PCR. Okay, why they are using, what is the main functions? If you want to a particular protein molecules, for example, mRNA, particular messenger RNA encoded a particular protein molecule, we need a number of copies. Number of copies means not less than 1 million, 2 million, 3 million. Like that, if you want a particular exact copy of the DNA molecule, if you want to synthesize it, what is the use? PCR mostly used for polymerase chain reaction. For example, a particular nucleotide, 1 to 100, you need millions of copy, means we are using the technology is called as polymerase chain reaction. Likewise, the polymerase chain reaction, what is the use of the particular protein molecules also synthesized correspondingly, because it encoded particular genetic code. For example, a copy of replication process, after finish it, a number of copy 1000 replicated, DNA molecule obtained means the DNA molecule further used for the, what is that? Translation process, particular protein molecule. Already you know what is meant by polystyrene chromatin. Chromatins, you know, are different types. Special type of chromatin, will you please tell me what are they? Polytene chromosomes. Already studied or not, media? Any idea, Dhanalakshmi?
No, sir.
What about Varsa?
No, sir.
Okay, anyhow, so whatever it may be, just go to the a special type of chromosomes. Special type, polytene chromosome, particularly present in the one of the worm is worm. Is nothing but the just like a ringworm, and likewise one of the worm is also present in it. That worm's salivary gland present polytene chromosomes. The polytene chromosome only present in the salivary gland of the particular worm. I need the worm name next class. Okay. Because what is the use of that one? You know, similar to this one, the DNA molecules produce the exact copy of the DNA. It may be more than 1,000 and 100, mean the particular protein molecules only synthesized by this DNA molecule for their growth, development, body development, because that particular chromosome mostly present in the young stage of larva only. That is the reason, because it's a growing stage, you know, so they need more protein molecule. That's why that type of protein, polytene, okay, the chromosome, Lamprey's chromosomes, that chromosomes are present in the larva stage. So they can synthesize more number of DNA, they can synthesize more number of protein molecules, then only the worm will be developed very quickly within short period. Okay, so anyhow, just go through it just for the information only. Here, the DNA replication is nothing but it undergoes the, what is that? Produce the exact copy of the DNA molecule of the parent. See that? So what are the stages of the replication process? There are 3 stages have been reported. Already you know about. Any one of the process, there is initiation is there. Okay, so the initiation. Take an example, protein biosynthesis. Which enzyme is required? Which codon is required for the protein initiation? Which codon, my dear? Triplet codon essential for the, otherwise called a starting codon essential for the protein biosynthesis. Will you please? Which codon? Starting codon and stop codon is also there. There are 64.
Sir, AUG is the starting codon.
Very good. Exactly correct. Exactly. Okay. So will you please, Dhanalakshmi, once again, that AUG is also mentioned a particular amino acid. Can you? That is very, very important. Okay. Do you know?
No, sir.
Okay, because sometimes they may ask the question related to codon. Sometimes they may ask the question related to the amino acids. Okay. So what about the Varsa? Do you know? AUG is the starting codon, correct? So what about the amino acids? Any idea? AUG? Do you know, Pa? Whether no or no, unknown. Sorry. Please.
Hello,
Answer
sir.
it.
Am I audible?
Pardon, Pa?
Sir?
AUG is denoted as which amino acid?
The methanol, something like that.
Sorry?
Sir,
Methanol.
Methanol?
methionine.
Yeah, very good. Who stated? Who said that? Dhanalakshmi.
Yes, sir.
Good. Methionine is the starting codon. Don't forget. Okay, this is very, very important one, my dear students. Sometimes they may ask the questions. How you know? So, amino acids aspect, sometimes they may ask the questions, which one you know? The codon aspects. AUG is your starting codon. It is encoded the amino acid methionine. Okay. So starting is methionine. Sure. Okay. Here, what are the stages of the replications? There are 3 stages have been reported. One is called as initiation, elongation, and also termination. See that initiations. The DNA synthesis is initiated at a particular point within the DNA strand known as origin. Simply, the particular origin, what happened, the replication takes place. That origin is also called as ORI, O-R-I, okay, origin, which are specific coding regions. Already you know about. It is also done by some of the enzymes. Once again, initiation is a particular point within the DNA strand known as origin, which are specific coding region. These origin are targeted by initiator protein, which go on to recruit more protein that help aid to the replication process, forming a replication complex around the DNA origin. There are multiple origin sites, and when replication of DNA begins, these sites are referred to as the replication fork. Suppose if it is not started at any point, it started any point of the protein molecule, but particular origin only, but it's not a single origin. There are so many origins also replication takes place. So that is the initiation process. Okay, the initiator protein is also very, very essential for the reactions, action of the initiation, particularly origin. A particular origin is responsible for the, what is the replication process. That is called as replication fork. Okay, so see that one. Within the replication complex is the enzyme DNA helicase. Don't forget, which unwind the double helical and expose each of these 2 strands. You know all about the DNA molecule. How many strands is there? 2 strands. Okay, these 2 strands, how it will be separated? How it will be separated? With the help of the enzyme. That enzyme is called as what is known DNA helicase. Don't forget, my dear. Which unwind the double helicals and exposes each of the 2 strands, so that they can be used as a template for replication. This is the first starting. It does this by hydrolyzing the ATP used to form the bond between the nucleobases, therefore breaking the bond holding the 2 strands together. Already you know, but DNA molecules are constructed or condensed with the, what is that? 2 components. What is that? Nitrogen base as well as a sugar molecule, isn't it? In between 2 nitrogen bases, there is a bond is there. That bond is what happened, breaking the bond, holding the 2 strands together. It can be divided with the help of the ATP molecules. Here the energy requiring process also occurs. Does this by hydrolyzing the ATP molecule. So energy requiring. So not only that, the 2 DNA strand is also separated with the help of the DNA helicase. Not only that, and also the presence of ATP molecule. As a result, you will get the 2 strand. In between there is a, what is that? Bond completely break. Okay, see that one. So next, DNA primase. So, another enzyme that is important in DNA replication. One is helicase, another one is a DNA primer. DNA primase is also synthesized with, it synthesizes a small RNA primer. DNA primase synthesizes the RNA primer, which acts as a kickstarter for DNA polymerase. Sometimes they may ask the question. DNA polymerase is also acted by one of the RNA component. Which one? RNA primer. The DNA polymerase is the enzyme that is ultimately responsible for the creation and the expansion of the new strand of the DNA. So you must note it down. So just like that, which one? AUG is a starting codon in protein biosynthesis. Likewise, RNA primer is used for the DNA replication process. That is very, very important. It is a starting point. The origin, the genetic code starting point. Okay, the helicase is, what is that? The bond break is taking place. DNA primase is also essential enzyme. It is also synthesized RNA primer. The RNA primer, what happened, which act as a kickstarter for DNA polymerase. Then only the polymerase enzyme, what happened now? Creation and expansion of the new strand. It can move on to the DNA molecule and also it produces a exact copy of the DNA. This is very, very important. So don't forget. So elongations. Once the DNA polymerase is attached to the original unzipped two strand of DNA. Once the DNA polymerase attached to the, what is that? Unzipped two strands of DNA, it is able to start synthesizing the new DNA to match the templates. I will show you in the diagrammatical aspect, you can easily understand. It is essential to note that the DNA polymerase is only able to extend the primer by adding free nucleotide to the 3' and sometimes they ask the question, DNA polymerase is only able to extend the primer by adding a free nucleotide to 3' end. This is very, very important, my dear. Okay, see the diagram, you can easily understand. 1. Now the template is read in the 3' to 5' end direction, which means that the new strand will be formed in a 5' to 3'. Here, starting from 3' to 5'. Okay, so opposite direction, what happened? The new strand will be formed. Okay, don't forget. This newly formed strand is referred to as a leading strand. It is continuous strand. There is no any breakage. From where to where? 3' to 5'. This is very, very important, my dear. 3' to 5'. I will show you. Okay, the diagrammatical aspects. Then see that along this strand, DNA primase only need to synthesize an RNA primer once at the beginning to initiate DNA polymerase. This is very, very important. That means during the replication process, two strand is there. Okay, DNA polymerase once act upon the DNA molecule. What happened? Only one primer is enough. Which strand? Leading strand. Only one primer is enough. Which RNA primer? RNA primer is enough. The opposite strand, what happened? They need each and every step. What happened? The RNA primer is essential. Okay, see that? This is because DNA polymerase is able to extend a new strand DNA by reading the template 3' to 5', synthesizing in a 5' to 3' direction as noted above. See that one? Diagrammatical means you can easily identify. See that one? So this is the structure of DNA replications. Okay, so DNA replication here. Okay, see, this is called as a leading strand. They are telling it which one isomerase, topoisomerase, primases, helicase. DNA helicase, what happened? It completely what happened at the bond breakage between, just like a zipper-like fashions. Zipper-like means you can split the molecule into two. Okay, the primase is the starting one. Already you know it is synthesized by the DNA primase or polymerase enzyme. See that? RNA primer is the starting one. Okay, opposite side also there is a RNA primer is there. This is leading strand. This is leading strand. Continuously it synthesizes the molecules. Only one primer is enough. Here the only one RNA primer is there. It leading to the strand continuously. But opposite direction, what happened? There's an each and every time there is a RNA primer is needed. That stage is called as lagging strand, discontinuous stage. When the DNA polymerase move on to the DNA molecule, each and every origin, there is a RNA primer is essential. Here the RNA primer is and here also the RNA primer, and another one is also, it comes in this area. Okay, So this condition is called as what is it? This fragment. Each and every one there is a fragment, small bit like that. So that is called as what is an Ogasagi fragment. Okay. So the replication process is, see here, starting from the 3' to 5'. Here the 5' to 3' there is a primer. Each and every replication process. What they need, RNA primer is needed. Here the one primer is there, another primer is also another primer. But in this category, only one RNA primer is there in leading strand. Lagging strand, each and every step, RNA primer is essential. Okay. So at the same time, it's a discontinuous strand. That strand is called as Ogasagi fragment. It's a small bits. Okay. This is the parental one. Okay. These are all are very, very important. This is the process. See that? The same things is also again. See, the replication started here. The replication fork is here. Okay. So replication fork. This is also replication. Starting from the 5' end, started from the 3' end. Here 3' end and 5' end. See the direction. See the direction. So this is started from this. This is the way, isn't it? This is the way. This is called as leading strand. Leading strand meant there is no any discontinuity in this area. Okay. What about lagging strand? Each and every time they need which one? RNA primer. The three green color represented RNA primer. RNA primer. Okay. So these are the, what is that? Lagging strand. Opposite, there is a same process also. Lagging strand here also. Leading strand, it's a continuous, what is that? Replication takes place with the help of the enzymes. Already you know well about RNA primer. Ori, starting place in this area. This is called as ori. This is starting place and also leading like this. Here the starting place. This is also ori. Starting like this. It is a leading strand. This is also leading strand. Only one primer is enough. But lagging strand, what happened? There is a each and every step they need RNA primer. Okay. You started from where 3' to 5'. 3' to 5'. Opposite manner is also same. Okay. See that one? It's very clear. The replication occurs in both directions along all the chromosomes. And both strands are replicated simultaneously. That is very, very important. The discontinuous strand or the lagging strand is the one in which 5' to 3' synthesis proceeds first in the direction opposite to the direction of the fork movement. In bacteria, the Ogasagi fragments are 1000 to 2000 nucleotide long. See that? The Ogasagi fragments means what is the discontinued fragments? Okay. How many nucleotide means 1000 to 2000 nucleotide long, is mostly found in the prokaryote. In eukaryotic cell, they are 150 to 200 nucleotide long only. So this is also some differences between prokaryote as well as the eukaryote, in where the Ogasagi fragments only. How many nucleotide present in it? Sometimes they may ask the questions. And also one more is the replications start from where to where, 3' to 5'. Okay. And another point is 5' to 3'. Replication bubbles form at the origin. The bubbles grow as a replication fork, move along the DNA in both directions from the each origin. Eventually, individual bubbles meet the fuse. Yeah, Y-shaped structure forms when a replication fork reaches the end of the DNA molecules. This is very, very important. Y-shaped. When all bubbles have fused, replication is complete and the two daughter molecules separated. DNA replication proceeds bidirectional from the origin of the replications. See the structures, you can easily identify. See that? So that one is different compared to this. This is the correct way. You can easily identify. Okay. See, first of all, you should know the DNA is a structure. It has a two strand. One is started from the 5' end. Another one is 3' end. Okay. So here, the 5' opposite, there is a 3' end. This is also 5' end. Okay. These two are opposite. These two are opposites. Okay. So The leading strand start from where, 5 dash to 3 dash, because here it connect with the 3 dash, isn't it? 5 dash to 3 dash. Only one prime molecules, the RNA primer is essential. It's enough for the leading strand. Okay. At the same time, they started from the middle portion. RNA primer is, see here, this is RNA primer. The arrow is RNA primer. This is lagging strand. Start up from here and finish it up to this level. And the another primer is also essential for the another origin and also leading to the 5 dash end. So these are lagging strand. These are leading strand. There is no any gap. This is continuously replicate until the reach of where 5 dash. Here in this point 3 dash we have to mention. Okay. Likewise, it is also lead to like this 3 dash to 5 dash, this part. So automatically what happened? It will be divided into 2 parts. This portion is called as 4 progressions. Hereafter the replication it will be stored. Here the helicase enzyme is there. Okay. The helicase enzyme, what they will do? The in between the bond it will be broken. In between the bond will be broken. The leading strand automatically it lead to the 3 dash and also lagging strand is also simultaneous. What happened? This is the one direction, this is another direction. Automatically what happened? It will be separated into 2 different copies. Okay, see that one. This means the replication proceed in the opposite direction away from the origin. The DNA synthesis is initiated by using a short stretch of DNA primer that is base paired with the DNA template. Already you know about it. What is the meaning of this one? Why we are adding the RNA primer means RNA primer is encoded some specific. What is that? Codon. The complementary with the parent DNA here. Parent DNA. So then only the origin, the replication process started. Okay. Likewise. Okay. So in each and every process also carry out. DNA synthesis initiated by using a short stretch of the RNA primer that is based parent with the DNA template. The size of the primer differ according to the initiation events. Sometimes the RNA primer it contain 2 to 5 nucleotide, sometimes 5 to 8 nucleotide. Sometimes it may be more than 8 nucleotide also present. The RNA primer is also, it has a specific, what is that? Codon. Okay, when you are going to study the PCR, their initiative codon is also added. That is called as RNA primer. That RNA primer commercially available. Just to go and check by any one of the genetic lab, you will get RNA primer. And also corresponding codon also they will give. These are the corresponding codon. So when you are introducing the newly discovered DNA or whatever it may, newly obtained DNA, it will be easily attached with the, which one you know? The corresponding ORI. Okay, see that one? In E. coli, the length is typical from 2 to 5 nucleotide. In eukaryotic cell, it is usually 5 to 8 nucleotides. Sometimes they may ask the question, RNA primer differ in their size. In prokaryote and eukaryote. Prokaryote, how many nucleotide? 2 to 5 nucleotide is there. Suppose if it is E. coli, sometimes they may ask the question, prokaryote. Sometimes they may ask the question directly, E. coli. They mention the name of the organism also. E. coli contain how many nucleotide? 2 to 5. And eukaryotic, 5 to 8 nucleotide is a primer. Okay. The short stretch RNA provides a primer onto which the DNA polymerase can add deoxynucleotides. Okay. So this is already you know a little bit. When you are adding the DNA polymerase, and not only DNA polymerase and addition with the RNA primer. These are the 2 very, very important ones. So DNA polymerase, what they will do? The DNA polymerase, what they will do? They will leading the strand. Polymerase enzyme essential for the DNA leading process as well as the lagging process also. Without DNA polymerase, the DNA it will not synthesize. That is very, very important. Okay. RNA primer, suppose if it is absent, what happened? The starting process, it will be suffer. Without starting process, what happened? DNA replication completely what will happen? Was out. It will not proceed. So these two are very, very important. At the same time, DNA helicase, what happened at the breakage of the bond? Okay. So DNA primer, helicase enzyme, DNA polymerase enzyme. These are the very, very important one, particularly RNA primer also initiator. Okay. So first, the replication process started by any one of the origin, RI, okay, RI gene with the help of RNA primer only. Without RNA primer, what happened now? The leading strand as well as lagging strand completely, what happened now? They will not proceed their work. Okay. See, the RNA polymerase that produce the RNA primer of DNA synthesis is called as permease. Once again, RNA polymerase that produce the primer of for DNA synthesis called permease. Okay, these are the things. In the 1960, Riji Okazaki, the colleagues found that one of the new DNA strand is synthesized in short pieces, now called as Okazaki fragments. Already we know about the lagging strand. It contains, there is a gap, isn't it? This is called as lagging strand. Each one, it has a small bit-like structure. Okay, it is not continue. How it will be joined, one of the enzyme is called as, what is that, Anybody else? Ajay? The lagging strand are united with the help of the enzyme. Dhanalakshmi? Varsa?
So DNA ligase.
DNA ligase. Ligase is nothing but the linking of two strands. Very good. Exactly correct. Okay. So excellent. So the start stage, already know about the Okazaki fragments. The Okazaki fragments, how it will be united, see that this work ultimately lead to the conclusion that the one strand is synthesized continuously and the other discontinuously. The continuous strands or the leading strand is the one in which 5' to 3'. Leading strand always start from 5' to 3'. Synthesize. Proceed in the same direction as the replication 4 movement. This is very, very important. Okay. See that point. So this is the already we know well about the leading strand completely started from the 3' end to 5' end. Okay. So DNA helicase, what happened now? Completely breaking of hydrogen bands. Okay. This is the RNA primer. So here also the RNA primer is there, but at the same time, what happened? Starting from this area, here too, and again here start from. Okay. There will be DNA polymerase leading. Here also the DNA polymerase. This is the RNA primer. Here also the RNA primer. Likewise, it may be continued. So the discontinuity, how it will be form a leading strand means with the help of the enzyme DNA ligase. DNA ligase means Okazaki fragments connected with or linked with the enzyme is called as what is that? DNA ligase. Don't forget, my dear. This is already so many times they were asked the question in NEET paper. 3 times. The lagging strand connected with the which one of the enzyme polymerase ligase, DNA ligase, like that they may ask the question. RNA ligase. Sometimes suppose they may ask the question likewise. Okay. So this is very, very important. Finally, what happened now? It will be completed from started from here to here, and it is also started from here to here. From this, okay, see that this part is this strand is mother strand. This is newly formed one. Likewise, opposite side, this is also a newly formed strand. This one is mother strands. This one is complement to this one, complement to the newly formed strand. So likewise, this is the parental strand. This is complemented to each other. So finally, what happened? These two are separated, and also this strand also separated. You will get 2 copies. Again and again, it repeated means you will get more than 100,000. That condition is called as what is that process is called as polymerase chain reaction. Chain means it's continuously. The replication process also done. Okay. As a result, you will get number of copies. Okay. Anyhow, the enzyme protein involves in the DNA synthesis. This is another one. The entire complex responsible for the coordinated DNA synthesis at the replication fork is a replisome. Okay, replisome. This is also another important things. See here, protein and function. DNA polymerase, what they will do? Deoxynucleotide polymerizations. Helicase, what they will do? Unwinding. Unwinding means zipper like patient, already you know about it. Zipper means the zip, what they will do, the two, what is that? Zip, stand like that, imagine, stand like that. Center, it will be remove the contact between the two zip. Okay. DNA chirase, otherwise called as DNA topoisomerase. This is very, very important, my dear. Relieve torsional strain that result from the helicase-induced unwinding. DNA primase initiates the synthesis of RNA primer. Already you know DNA primase is essential for the synthesis of RNA primer. RNA primer is very, very important for the initiation process of the replication. The single standard binding protein. This is also given in your slide. Prevent premature re-annealing of double stranded DNA. And DNA ligase. What is meant by DNA ligase? DNA ligase catalyzes the formation of the phosphodiester bond between the 3-hydroxyl at the end of the DNA strand and the 5-phosphate of the end of the. That means DNA ligase mostly used for the, what is that? Connection between two things. What is that? The Okazaki fragment. One side 3' end is there. Another strand, it has a 5' end. The both are united. Okay. That's why they are used in this. So these are the, so protein. Why it is? These are all are enzymes. Don't forget, my dear students. These are all are enzymes. The enzymes all are made up of which one you know? Protein. Okay. So what are the functions? So don't forget, this is very, very important. See that enzyme in DNA replication. First one is helicase. Helicase, this is the enzyme. Okay. This enzyme, what they will do? Completely the bond breakage. Okay. So another one is binding protein stabilize separate strand. This is the binding protein. And another one is primase added sort primer to template strand. This is the ORI. So we are adding the, which one you know? Primase enzymes. Okay. DNA polymerase 3 bind nucleotide to form a strand. This is very, very important. DNA polymerase 1. That is also a exonuclease. This one is called as endonuclease. This one is called as exonuclease. Remove RNA. Okay. So this is the other parts. Ligases. After the Okazaki fragment, when we obtained the end of the reaction, what happened? You know, the RNA primer. Okazaki fragments, we will get it, isn't it? The Okazaki fragments join together, then only you will get the double stranded. Otherwise, what happened? You know, it will be lack of strand. Okay, so ligase join Okazaki fragment and seal other nick in the sugar phosphate backbone. So these are the enzymes are involved in the replication process. Here they may ask the question. These 4 points is very, very important. Helicase, DNA polymerase 3, DNA polymerase 1, and ligase, DNA ligase is very, very important. Already you know very about the DNA primase is used for the synthesis of DNA, RNA primer. DNA primase used for the synthesis of RNA primer. Okay, so this is our important point here. So this is the very, very important. See here. So same things is also given here. Helicase, unwinding DNA. DNA chirase relieves the stress created by the unwinding. As proteins bind to the single stranded DNA and stability it. Primase, synthesis of RNA primer. DNA polymerase 1, excision of primase and filling the gap with the nucleotide. DNA polymerase D, new strand elongation. Very, very important. New strand elongation, which DNA polymerase involved are essential. Mean you have to tell DNA polymerase 3. And DNA polymerase 2 also is there. The DNA polymerase 2 is also present in it, but when the both enzymes, DNA polymerase 1 and the DNA polymerase 3 is not activated, that time DNA polymerase 2 activate on it. Don't forget, my dear. DNA ligase is also there. DNA ligase only join the Okazaki fragment corresponding join. Then only we'll get the 2 copy of DNA molecules. This is very, very important. Okay. See that one. Polymerase is also there. DNA polymerase 1, polymerase 2, polymerase 3. Okay, the mammalians alpha and the epsilon E. Okay, beta, gamma, delta. It is a Roman letter. Okay, so what are the things is that lagging strand synthesis, gap filling, proofreading repair. Proofreading repair is also mitochondrial DNA synthesis. This one is also another part. So these are the mechanism of DNA replication. What is the main mechanism? What happened? All living cells such as E. coli replication require a set of enzyme. First one. Any one of the animals, whether it may be a prokaryote, whether it may be a eukaryote, okay, they need a particular set of enzymes. The enzymes, without the enzyme, what happens? Any reaction, it will not happen. Take an examples. Glucose convert to glucose 6-phosphate in glycolysis. Without enzyme, it is not converted. Take an examples of curd. Lactose convert to lactic acid with the presence of the enzyme is called as lactase, lactobacillus. Okay. Likewise, the enzyme is very, very essential. Here you must note it down. How many minutes it will take each and every organisms, particularly lower animal E. coli, complete the replication of its DNA in within 38 minutes. Don't forget, my dear. Don't note it down, my dear. This is one of the questions. I think 2019 or 18, this question asked in NEET. In E. coli, the DNA replication will be completed 38 minutes exactly. The average rate of polymerization has to be approximately 2000 base per second. Per second. 1 second, how many base pair it will be polymerization takes place within the E. coli means 2000 base pair. Note it down. The polymerization process must be accurate. By mistake during replication would result into the mutation. Already told you. The replication is very fast. 38 minutes, it's enough for the replication completion process in E. coli. Per second, how many base pair it will be polymerized? Mean 2000 base pair polymerization takes place within the E. coli. Suppose any difficulties, suppose if it is take an example, here the 38 minutes is the, suppose if it is it will take 48 minutes, means 10 minutes more than that, means what happened? Something happened inside the nucleus base, polymerization reactions. So it may be due to the, what is that? Mutation. Mutation means the sudden changes takes place within the chromosome or any other mistake. Sometimes man-made mistakes also done during the replication process. Okay. As a result, what happened? It will lead to some difficulties. Okay. That is mistake during the replication would result into the mutation. So, okay. So here you must note these 2 points is very, very important. This one is also. Okay. Without enzyme, there is no replication process. 38 minutes, we will have to finish it. Finish. And how many base pair is very, very essential. Suppose if it is difficult, what happen? Lead to the mutation process. See here, already we know about deoxyribonucleoside triphosphate. What are the types of deoxyribonucleoside triphosphate is there? See that AMP and GMP, CMP, TMP. What is that? These are all nitrogen bases. Monophosphate is added. Deoxy adenosine monophosphate, simply called as DAMP. Okay. And also deoxyguanosine monophosphate. So DGMP. DCMP, deoxycytidine monophosphate. And the last one is TMP, deoxythymidine monophosphate. Serve dual purpose. Already you know about ATP is essential for the replication process. Already we studied. It's also very, very important in starting. Okay. So they are all providing high energy compound. That is very, very important. See that provide energy for polymerization and act as a substrate for polymerization. They need some substrate. Suppose any one of the enzymatic activities occurs, they need some substrate. Without substrate, they will not proceed the reactions. It is also one of the enzymatic reaction, similar to that of the enzymatic reactions. Okay. So these are the high energy providing component. Okay. So this is very, very important. So and then next, there are so many enzymes there. What is the main role? Each one, it has a specific role is also there. Okay. The role of topoisomerases. Role of topoisomerases. Double stranded DNA is unwound by helicase. The unwound activity of helicase at one position along the DNA molecule causes the downstream effect. Also done by which one of the following enzymes? Sometimes they may also call downstream effect. So helicase, otherwise called as topoisomerases. Okay. Portion of the same DNA molecule to become a overwound, unless this overbinding is relieved by the enzyme called gyrase, topoisomerases. Okay. Suppose the helicase enzyme is over, what happened, unless this overbinding is relieved by an enzyme called gyrase, DNA replication will soon stop. Okay. So the unwinding is what happened when it will be stopped with the help of the enzyme is called as gyrase. The gyrase use ATP energy to introduce negative supercoiling into the DNA. This is another point. Gyrase is also uses ATP, high energy compound, introduce negative supercoiling into the DNA. The gyrase can be regarded as a swile for replicating molecule. This is another point. Simply, you just note it down. Topoisomerase, helicase enzyme, downstream effect. Suppose if it is extended, what happens? Automatically the gyrase is secreted. So the DNA replication stop. Okay. The gyrase is also otherwise called topoisomerase. What happens? It is without energy, they will not proceed their function. So they need energy in the form of ATP. Okay. Got it? Okay. For the next one. So this is the things. The schematic mechanism of action for the topoisomerase 1. Likewise, there are so many enzymes there. See here, the cut is made, neck, followed by boss stand. So the bubbles like structure is also formed as takes place. Again, it will form as a N+1 stand. Simply schematic. Just go through it, you will get it. So this is another point. The same one. See the started from here like this. The leading. Finally, what happened? Okay, topoisomerase. The various steps involved in this process are summarized as follows completely. What are the steps involved? Okay, we will finish it up to this level only, I think. Mechanism of replication start as a specific point of the DNA molecule is called as origin or ori. They may ask the question, replication started in which place? Ori, origin, or origin. The origin DNA strand break because of an incision nick. This is made by the enzyme is called as incision enzyme, otherwise called as endonucleases. Okay. And the hydrogen bond join the two strands are broken by the enzyme. The endonuclease is called as what is that? Helicase. Okay. So already told you, helicase enzyme. The stone strands starts unwinding enzyme, helicase. The two polynucleotide strands are thus separated. Okay. So this is the process. The point where the two strands separate appear like a fork of Y shape. This is unwinding part. This is the Y part, this is the Y-shaped nature. When we are seeing the Y shape, that is called as, what is that? Fork stand. This is described as a replicating fork. This part is replicating fork. Already you know about. The new stand is constructed on each old stand. New stand is also constructed on each old stand. Already told you. Parental stand is the followed by the complementary gene is new stand. Likewise, opposite side, there is a old stand is the followed by there is a new stand is also constructed. Okay. This takes place with the help of the small RNA primer molecule, which is complementary the DNA at the point. Already know. Each old DNA stand acts as a template for the construction of new stand. Already you know about that things also. The RNA primer attaches itself to the old stand and attract the enzymes DNA polymerase 3. Okay, this is very, very important. For example, I will show you. Okay. Okay, stop. See here. So listen, my dear. See, so this is considered as a DNA strand. So this is considered as a DNA strand. Here 3 dash and here 5 dash. Why I am telling you know, this one is called as 5 dash and this one is called as 3 dash. Okay. So here in between what happened, you know, there is a, so I have to draw like this. Okay, I have to draw like this, then only you can understand structure. So in this part, here what happened, the RNA primer is there, isn't it? In this part, there is a RNA, okay, primer, isn't it? RNA primer. Okay, so it is started from here to standing like this, RNA primer. So how it will be, you know, here there some of the codon is also present in. Codon means already in our board, adenine, okay, and guanine. So suppose take an example, AUG is a starting codon like that, AUG. Okay, AUG is a starting codon like this, no? Okay, so the corresponding, what is that? Corresponding. So, okay. Okay, here AUG now, think about this one, AUG, imagine. Okay, the opposite side, what happened, you know, the correspondingly A is always combined with the molecules of thymine, isn't it? Okay, but in this category, RNA molecules, A combined with the molecule of U. And here, so U combined with the molecules of A. Here's G is the, it combined with the molecules of C. Guanine combined with the molecules of cytosine. And uracil combined with the molecules of adenine. Adenine combined with the molecules of uracil. These are the part, isn't it? This part mostly present where, you know, in the DNA molecule, in this part. DNA stand is here. This is U, A, C. So the RNA primer already in our, but this is the component, AUG. So this is the starting, AUG, AUG. So correspondingly, so here RNA polymerase is also there. This leading strand is also going on like this. Okay, so opposite side, what happened, you know our board like this, in this part, it's going on like this. So in this part, what happened, started from here, there is a lagging strand like this. Here the topoisomerase, what happened, completely unwinding the bond. At the same time, each and every origin, there is a RNA primer they need. So RNA primer. Okay, these are called RNA primer. This is going this way. Okay, so finally what happened, the stool stand will be completely, you will get it. Stool stand, likewise, you will get it. Okay, in this stool stand, the first one is there, this is the parental stand, isn't it? This one. So this one is the parental stand. Okay, this one is the newly formed stand. That means the center inside there is a which one? This one is what is a newly formed stand. This one is a new stand. Okay, so new one. So likewise, this one is the parental stand. This one is the new stand. Okay, so new stands. So what they are telling, this parental stand is the complement to the this one that is given here. That is given here. Okay, so this is the parental stand. It has some codon. Okay, it is complemented to the newly forming stand. Newly forming stand which encoded the AUG. Okay, then only the DNA molecules replications also takes place. Otherwise, what happened? It will not started. The RNA primer and started at the same time. It complemented to each other. It complemented to each other. Okay, so this is very, very important, my dear students. Don't forget. So here, what happened? This one is 25% of parental stand. This one is also 25% of the parental stand. The center is a new stand, isn't it? These are the 25%. This is also another Okay, this one is also 25% of newly formed one. This one is also newly formed one. Imagine this DNA molecules, and this one is also vary. This is similar to that of this one, isn't it? This one similar to that of this one. So this is also so total how many percentage, 25 25 25 and 25%. Okay, these two are new strand, these two are parental strand. Don't forget, my dear, this is very, very important things. Okay, okay, see here. Okay. Okay, this one. Yes. Okay. So this is the pointer. So that thing's already given here. So already I mentioned what is my leading strand, what is my lagging strand, how it will be forms. See the structure, entire structure. This is called as a RNA primer, followed by this is the lagging strand. Okay. This one continuously forming strand, that is called as leading strand is there. Okay. This is the structures. Finally, you get the two strands. The replication of the lagging strand takes place in the small fragment is called as Ogasaki fragment, already you know about. These are then connected together by the enzyme ligase, DNA ligase. Replication may take place in only one direction on the DNA helix, unidirectional, or in two directional, bidirectional also. At the end of the process, two double-stranded DNA molecules are found from the original DNA molecules. This is called as replications. Okay, so remaining things, mutation, we will see the next classes. Today, what we'll discuss, will you please completely summarize all those things? What is by DNA replication? How it will happen? What are the enzymes also involved? Okay, helicase, topoisomerase, and also RNA primer, DNA primase, and also helicases and ligases. Okay, how it will be started from the 3' to 5' and 5' to 3'. Unidirectional, bidirectional, whatever it may, and also which enzyme is involved, and how it will be initiated. There is a particular origin is called as origins, and also started from the 3' to 5'. There is a leading strand, and 5' to 3' is a lagging strand. So each one it has a specific RNA primer they require. Then only the lagging strand is continue. Finally, what happened? It will be produce a new strand. Okay, two exact copy of the DNA will be formed, particularly the E. coli molecules. E. coli. Okay, how many minutes it will take? So, 38 minutes. Don't forget, my dear students. How many nucleotide per second mean 2000 base per nucleotide polymerization takes place. It is very, very important, my dear students. Okay, so don't forget, my dear students, hereafter. Okay, we'll see the remaining things on the next classes. Please follow it. It is very complicated subject. At the same time, how many questions they will ask in this part? Maximum question 12 to 14 questions. 12 to 14 questions in these two parts: molecular basis of inheritance and also principles of inheritance. These are the two important chapters. Is the maximum question 12 to 16 questions may ask. Okay, don't left. Okay, thank you so much for the participation. Thank you. Any question, my dear?
No, sir.
Okay, okay. Okay, Ajay, thank you. Thank you, ma'am. Thank you, Varsa. Thank you, Dhanalakshmi, and so on.
Thank you, sir.
Thank you, Pa.
Thank you, sir.
Thank you, ma'am.